Hello children, good morning. Hope you all are doing good. Welcome to the program NEET Foundation video for part 4 video. Up till now we have completed the introduction part of the cell division and cell cycle and the different types of cell divisions like amitosis, mitosis and meiosis. Okay, and in a previous class we have discussed about what is the interphase, right? So what are the roles of interphase in cell division process. So in this video we are going to discuss about, especially in the interphase, what are all the phases which are involved in the generation, producing the next cells, nothing but the daughter cells, how they are being produced either in the mitosis or in the meiosis. So in general we can discuss any one of those two, but in this concept we are going to discuss about the type of phases which are present in the, which undergoes in the cell cycle or a cell division. So let us see what is the first concept, the third phase. The first phase is the G1 phase. As we discussed already, majorly we can observe the 4 phases in the interphase. First one is the G1 phase, gap 1 phase or the growth 1 phase. Second phase is the S phase, synthesis phase, or also called as a replication of DNA. And the third phase is the G2 phase, gap 2 phase, or also we can say it as a growth 2 phase. And fourth one is beginning of the daughter cells dividing into mitotic cell division. Let us see the first concept of the G1 phase. This is the first phase in the interphase, where it is being seen in the interphase, it is the first phase. So from the end of the previous phase till the beginning of the DNA synthesis is in the next cycle is called as a gap 1 phase. So here G indicates gap, or also we can consider it as a growth. So this phase is also called as growth, right? So let us see the picture what is given over here. If I am very clear to this, so here you can observe the half partition of the cycle is whitish in color and half part is somewhat like pinkish in color. Okay, so this part is the mitosis. This part is the mitosis and this white color one is the interphase. Yes? Yes, this is the interphase. This is a mitosis and this is the interphase. What is the G1? Gap 1 phase he is talking about. So this is the phase where we can observe it's previous to the M phase till the beginning of the DNA synthesis. Previous to the M phase. So within the M phase we can observe the prophase, prometaphase, metaphase, anaphase and the telophase. So this one occurs. So this as it starts, before to this we can observe the interphase, and as this finishes, after the telophase, the daughter cells, how many they are being formed, each and every cell is responsible for undergoing the interphase process. Okay, so this is a continuous process. So here we can observe that from the end of previous M phase, mitotic phase in the previous cell, and beginning of the DNA synthesis. Why the cell has to, why the DNA has to synthesize? It has to replicate, cut open, and the base pairs, the chemicals which are present in those must be segregated towards to the, released outside towards to the cell, so that the continuous mitotic or the meiotic cell division do occur, right? And this is the continuation phase. So next let us see the next phase. Next, continuation of the G1 phase. In this phase what happens? Okay, so there we can observe a type of activities called as biosynthetic activities. Synthetic means preparing, manufacturing something. What do they manufacture? So which shows a considerable slowdown during the M phase of resumes, it activates at a high rate, nothing but. So here we can observe the new cells are being forming. So these new cells are undergoing the interphase. Interphase is helping for the daughter cells to undergo the mitotic cell division. Single chromosome, condensed chromosome is responsible for dividing, doubling the number of chromosomes into new cells. Okay, that is called the interphase. So this in this we can observe the size of the cell will be increases. What is the difference between these two cells? This is a small cell and this is the large cell. Okay, so this is responsible for, this is based on what? The biosynthetic phase. So in the G1 phase, so because of this synthesis, replication of DNA, the chemicals which are released into that, the size of the chromosomes will be increased and the formation of cell organelles also can be seen at the end of the cell cycle, end of the mitotic cell division. Okay, so here the majorly we need to identify the size of the cell. Okay, so size of the cell will be increasing by the biosynthesis which is occurring inside the cell, right? Next, so still what happens in the G1 phase? So in this phase there is a marked production of proteins. As we discussed previously, the cell is getting ready to divide. So they are producing the proteins, it seems. How they are being produced? These production of the proteins are being done by the 20 amino acids. The chain linking system of the 20 amino acids and also catalytic products called as enzymes which speeds up the reaction, right? So here that are required for the S phase. So this enzymes and the protein productions with the help of a 20 amino acids, they are responsible for synthesis phase. DNA replication has to be done based on the proteins and also the enzymes. After the production of proteins, that has to be reacted with something within the cell itself. So it has to speed up the reaction. Okay, so how much time it takes? Within a fraction seconds. So this kind of DNA will be replicating and the duration of G1 phase is highly variable. Highly variable in the sense, in multiple cells. In our body only there are multiple cells. Each and every cell, the G1 phase duration will be completely different as per the requirement of the cell, how many they need to be produced. Okay. Let us see in the picture here. See this amino acids, fatty acids and the energy which is produced by the oxygen. So this amino acids, and see here in the replication process we are observing the DNA polymerase, helicase, ligase. Okay, so these are the different types of enzymes which makes the division of the cell, okay, which makes the replication of DNA and again the synthesis. Replicated DNA again will be going back to the spiral position. So what is the use of this leading strand? So here there is a continuous double helix structure. Here it got divided. Yes, so there is a open up process and again after some gap this fragment is called as a Okazaki fragment. So after to this, the enzymes, these are adding the nucleotide and synthesizes the new DNA. Synthesizes of the DNA will be done by the enzyme called as a DNA polymerase. And next one is helicase. What is the use of this helicase? So this enzyme catalyzes the hydrogen bond to unzip the double helical structure. So this bond, this enzyme is responsible for unzipping the double helical structure into single helical structures. Okay, and what is the next enzyme? Ligase. So it connects the fragments together via phosphodiester bond. Okay, so these are the different types of enzymes. Some are using for joining and some are useful for rejoining, in the sense it opening. Okay, so as it opens, the chemicals which are present in the base pairs which are present in this base pairs are the A, T, G and C. What are those? Adenine, thymine, cytosine and guanine. Okay, so these are the base pairs. They may change the sequence during the replication process and again the sequence will be same as they are ending the process of the replication. Okay, so further the three enzymes are used over here are called as the DNA polymerase, and next one is the helicase, last but not the least is the ligase. Okay, and this one is the continuous process where the cell is produced, new cell is produced by the previous cell. Next, what is the next concept? After G1 phase, we can observe the synthesis phase. So, this synthesis phase, the major event in the cell cycle is replication of DNA. So, this we must keep it in our mind. Until and unless the DNA replication is being done, the cell, the chromosomes, the nucleus, the cell organelles, the cytoplasm, whatever material present inside the cell, they cannot able to divide. Okay, so this plays a very major role. Right. So, what is that process called as a replication of DNA? So, which occurs in the S phase and separation of the duplicated chromosomes. Exact copies of the parental chromosomes which are present in the previous cell, same kind of chromosomes are forming in the next existing cells, next daughter cells. So, that is being maintained by the replication of DNA. In the previous slide, we have discussed that the three enzymes which are helpful for opening, releasing the chemicals and again becoming double helix. Okay, and the constituents of the cell which occurs in the M phase. So, after this we can observe the G2 phase and the continuation of the new cell division called as a M phase. Right. So, let us see here. The DNA replication haploid versus diploid. Haploid in the sense single number of chromosomes are present in the gametes. So, that is being done in the reproductive cells. And next one is the diploid cells. Diploid cells are nothing but the somatic cells. So, let us see here. Before the replication process, the chromosome is only single. Means only single chromatid is present without the centromere, and after that we can observe 2 chromosomes. 2 chromosomes with actually, if there is a centromere, we can observe the 4 chromatids. In the absence of centromere, there are the 2 chromatids. So, this diploid cells have 2 sets of chromosomes. One set come from the paternal and another set come from the maternal. Okay, so mom nothing but the maternal and the dad comes from the paternal. Okay, so here the 1 chromosome with 1 chromatid and 1 chromosome with 2 chromatids. After the DNA replication, this is before replication. Okay, so this is a role played by the replication process. Okay, next. So, now let us see what is actually happens in the S phase. So, this S phase, the DNA replication, so as they are releasing the chemicals, so why do they release? They are responsible for in the mitosis maintaining the equal number of divisions, equal number of chromosomes are being formed as like in the parental cell, daughter cells also have same number of chromosomes, and apart from that, in the S phase, the DNA replication, the chemicals and the enzymes and the type of biosynthetic reactions what occurs inside to that, and the proteins which are produced inside the replication of DNA, those are responsible for regulation, nothing but the controlling pathways. They can control, they can't keep on going increasing the number of the chromosomes, they will be maintaining the particular number of chromosomes in the daughter cells from the parent cell, and the initiation where it has to begin. After interphase, the mitosis begins. After interphase, the daughter cells, each and every cell will be undergoing either the mitotic cell division done by the somatic cells and meiotic cell division will be done by the generative cells. So, after the initiation and the completion of S and M phase, replication of DNA will complete itself. It has a particular time duration, and also beginning of the M phase, when it has to begin. After the synthesis will be completed, ending of the DNA replication if it is been done, then the M phase begins. Okay, and this ensures the genetic information and other cellular components are duplicated and divided equally between the daughter cells with each cycle. Within the cell cycle interphase, so they are responsible for controlling process. okay, and also regulating process and also the initiation process, when it has to begin, when it has to stop, okay. So that is been done of both the genetic information and cellular components, nothing but the cytoplasm will be, cell organelles will be, and the type of materials which has to be maintained inside the cell organelles is also been, everything is done by the synthesis of a DNA, nothing but the S phase, okay. And after that, maintenance of the number of chromosomes, it plays a very major role. There is no abnormality, there is no sort of more segregation of chromosomes or less segregation of the chromosomes is not been done in the mitosis cell division, okay. So it plays a very major role, one of the phase of the interphase, and this equally distributed between the daughter cells and in each cell cycle, okay, nothing but the cell division, interphase, okay. So see this is one of the great example we can take, the DNA replication. The base pairs as we discussed previously are the adenine, cytosine, guanine and the thymine. And let us see here the unwinding, nothing but its opening, okay. So double helical structure of the DNA, it opens, and as it opens, the coiled double stranded DNA molecule unwinds and separates into two strands. So here it is double helical structure, yes. So as it comes over here, it got divided, okay. So here it is a double helical structure, and here it became, this is also the double helical structure but not complete. And where is a single helical structure? These are the single helical structures. So why they become as a single helical structure? To release the chemicals, okay. So here the complementary nucleotide bases are being attached, okay. So this is the complementary DNA molecules where there is a requirement of attached molecules. So here the base pairs adenine always binds with thymine and guanine always binds with cytosine. So that is a correct pairing of the DNA synthesis. After the synthesis also this kind of sequence may occur. Sometimes it may repeat, like adenine, thymine, guanine and cytosine. After that again adenine, thymine and guanine and cytosine. So here you see the pink, blue, green and cream. Pink, blue, cream and green. Blue, pink, again we got blue and pink and blue, okay. So this is a repeated one. The changing of the sequence may occur after the finishing or during the synthesis ending of the DNA replication, okay. So that is being done in the unwinding process and rebuilding process. What is the next event which occurs in this DNA replication? Rebuilding, nothing but after opening the single helical structures, again they will be binding back into the double helical structures. Enzymes connect a nucleotide with the appropriate base and to the growing new strand as the base bonds with the exposed complementary base. So to be frank, simply we need to discuss about this. Previously we have discussed about the enzymes, the catalysts which are responsible for opening and closing, okay. So opening and closing, in between what happens? Releasing of the chemicals, okay. So that is the rebuilding process. Last but not the least is each of the single strand becomes a double strand and each identical to the cell's original double stranded DNA molecule. After the sequence is been, it may change, but as it is moving again to the ending of the DNA replication, again the single one will be adjoining into two spiral structures, double helical structures. So there again they are starting the sequence adenine, thymine, cytosine and guanine, adenine and thymine. The sequence will be continuous within the replication of DNA as the new daughter cells are ready to divide into the new division process, okay. So this is the capacity of a DNA replication. Now let us see the next concept. Still what happens in this? Cell cycle undergoes the checkpoint systems. What are the checkpoints? What are the checkpoints? Checkposts will say, right? So there we can observe the checking of the products where the traveling agents are moving from one place to another place. So they may take the products legally or illegally. Okay. So in the same process, in the S phase, the checkpoints plays a very major role. So these are acting as a regulatory pathways. Regulatory in the sense controlling. So they have a capacity to control whatever the kind of event, even it might be the positive event or it might be the negative event, it is being controlled by the checkpoints which are seen in the S phase. So, and that controls the order and timing of the transitions. Transitions in the sense also the checkpoints, different checkpoints in different phases of the cell cycle. So, of the cell cycle, the checkpoints also ensures that critical events, any dangerous events, either the cell damage or any cell organelles damages or any nucleus gets damaged. So, each and everything is being checked by the checkpoints in the S phase. So, such as replication of DNA and segregation of chromosomes are completed before the cell progresses further through the cycle. Until and unless the checkpoint is being done, the cell would not able to put its step forward to undergo the continue the process of a cell division. Let us see the checkpoints where we can observe. So this is a complete schematic picturization like a pie diagram. So here we can observe the first phase is called as a G1 phase, next phase is called as a S phase, and next phase is called as a G2 phase, and fourth stage is called as a M phase. So where we can observe the checkpoints here, near the G1 there is one checkpoint. So this red mark is nothing but one of the checkpoint. So during this checkpoint, what do the stage do? Okay, so here they check the cell size is particular or not, is it flexible or not, is it sufficient for the production of materials which are being maintained by the metabolical activities furtherly or not. Everything is being checked by the checkpoint near G1 phase, and also it checks the quality and quantity of the nutrients. Quality, how much amount they require as per the size, and quantity. Okay, quality in the sense what are the type of nutrients. Type of nutrients, how much, what we can say that a type of quality in the sense purity. Purity is, we can say the purified nutrients if they are being produced, so that the metabolical activities basically which occurs inside the cell may occur very properly. And next one are the growth factors like the enzymes or it might be the proteins, right? So are they in a proper condition or not? So these growth factors will be playing a very major role. Are these in a correct position or not are being checked by the checkpoint near the phase G1, and also they will check the DNA damage. Okay, so this plays a very major role. If the DNA damages, the continuation of the cell division will be stopped. Okay, so if it is in a correct condition, then the continuation of cell cycle or cell division will be done. Okay, and next to this, it takes some rest. G0 phase. Okay, up till certain time some of the cells may take rest, but some cells like liver cells, kidney cells, nerve cells, in our previous class we have discussed, some of the cells they permanently take the resting condition. Okay, so after taking rest they do undergo the synthesis process, and again after the G2 phase we can observe the checkpoint near G2 phase. So in this also they check the cell size and they check the DNA replication, and after that this is one of the checkpoint, and after that in the mitosis they will be checking once again. So what they checking for? Chromosome attachment to the spindle fibers. Are they attaching properly? If they attach, then only they can be divided in a proper condition, so that the equal number of chromosomes, nucleus, cytoplasm, cell organelles will be distributed towards to the daughter cells. So these kind of conditions are also being maintained in the S phase by the process called as a regulatory pathways. Regulatory pathways called as a checking points. One is near the G1 checkpoint, one is near the G2 checkpoint and one is near the spindle assembly checkpoint. Okay, so after finishing these regulatory pathways, then only the further daughter cells will be undergoing the mitotic or meiotic cell division. Hope you all understood. And coming to the next concept, still what happens in the S phase, the cell cycle checkpoints respond to the cellular damage. Okay, so previously we have discussed it that they'll check everything. Okay, so during the checking process, if it finds any sort of problems, consequences done towards to the cell or the nucleus or to the chromosomes or to the DNA replication, so they will be responding. Nothing but if we get any sort of cut over here, injury over here, do we leave like that? We can't. Okay, so what do? Immediately we'll do the first aid, right? So after the first aid, it may just cure within few minutes or few days. Okay, so the cell cycle checkpoints respond to the cellular damage by slowing the cycle to provide the time for repair. Okay, so they will be taking some gap after finding out whether there is a problem or not. If there is no problem, they'll continue the process. If there is a problem, they'll give some time duration. For what process? They'll go for the repairing process, it seems. After the repair, then only we can go further, right? If for example, we will be writing with this hand. If this finger got injured, what do? Can we write? Can we hold like this and can we write properly as like previously? We can't. Okay, so this must be treated. So after treating it, after treating finished, treating is been done, treatment is been done. So this finger is normal. Before the treatment, the finger is not supporting for this, coordinating with the other fingers to write. Okay, so we'll face problem. So we can't do our work properly. In the same process, the S phase also, the cell cycle checkpoints respond to the cellular damage and they take some time for repairing it, and it also induces the transcription of genes that facilitate the repair. So whatever the factors are responsible for repairing that. So what is the medicine for curing that kind of damage? Okay, so those kind of damage is been done by the treatment is been done by the transcription process of genes that facilitate for the repairing process. Okay, so here the checkpoints are also responsible for checking the DNA replication process, whether it is been going in a normal process or in a damaged process. Okay, so DNA damage checkpoint and DNA replication, DNA damage again checkpoint, they'll go for double time, checking for double time, then only they can confirm it. Then the cell will be undergoing, the DNA will be undergoing the replication process, and after this DNA damage checkpoint near the G2 phase and antiphase checkpoint. Antiphase checkpoint in the sense further it has a capacity to undergo the mitotic cell division or not, it will be confirming for the final stage, right? And spindle assembly checkpoint, again they will be undergoing for the checkpoint whether the chromosomes are ready to divide equally or not with the help of a spindle fibers. Okay, so see here we can observe the transcription coupled damage. Okay, so TCD, this plays a very major role. So these are the transcription coupled damage. Okay, so as it occurs over here, it got a cut over here. Okay, so this, the chain is being just adjoined over here, and again after repairing it got continuous, and again we got a gap over here, damage. Okay, so again the transcription coupled damage is been cured, and again there is a binding process, checking process, and the DNA replication is being continued. Understood? Right. And what is the next concept? Role of interphase in the cell cycle. Up till now we were discussing about the same thing. Let's recapitulate once again. Cell cycle checkpoints are surveillance mechanisms. Surveillance mechanism in the sense, so they are the machines continuously work out whether the checking is being done or not. After the checking process, the cells, the type of chemicals which are present in the cell, they will be helpful for undergoing the process of a cell division, and these mechanisms that monitor that will be guiding where to go, how to go and when to go and all are being guided. And the order integrity, nothing but all the kind of chemicals, all the kind of substances which are present in this cell is undergoing in a unity. Integrity, nothing but the unity. They are undergoing their own process. If I do this, I can't do the other work. Another may be doing the other work. Okay, so another may be doing the other work. It's a kind of teamwork. So the complete cell is not been undergoing the cell division by only the DNA, by only the chromosome, by only the nucleus or by only the spindle fibers. All together they must undergo their own functions so that the complete cell may undergo the mitotic cell division and it can produces the new cells. That is a teamwork. And fidelity, okay, nothing but accuracy. After completing the work, by the checking points, regulatory pathways, they'll check whether the work is being done properly or not. If there is any sort out, they'll be just removing that and repairing it and again confirming it for the twice and thrice, and then they are making the allowing the cells to undergo the further cell division. So these include the growth to the appropriate cell size. So this kind of mechanisms, what kind of mechanisms? Cell cycle checkpoints are the surveillance mechanisms. So these surveillance mechanisms which are done by the checkpoints are undergoing the monitoring process and ordering the integrity and fidelity of the major events in the cell cycle. So the replication and integrity of the chromosomes, replication and integrity of the chromosomes and their accurate segregation at the mitosis. Then only the mitosis begins until the checkpoints will do their complete work. Okay, let us see in the picture what is given. So the overview of the mitosis. Without the interphase, the mitotic cell division continues? It can't. Okay, so within the interphase we can observe the G1 phase, S phase, G2 phase and the beginning of the M phase. Okay, so then only we can observe the early prophase. We can observe the formation of spindle fibers and disappearance of the nuclear envelope. And here we can observe in the late prophase, the spindle fibers are completely attached, segregated towards to the cell and making the holding up function of the chromatids along with the centromeres. And next one is the metaphase. So within the metaphase they form the equatorial plate. Each and every chromosome along with the centromere will be attached towards to the freely suspending towards to the equatorial plate with the spindle fibers, so that equal elongation, division of the chromosomes will be done into the opposite poles in the anaphase. And last but not the least is the again the rejoining, reformation of nuclei, nuclear membrane, nucleus will be dividing into two nuclei, karyokinesis and also the cytokinesis. The complete energy, processes energy is being given by the interphase. So this interphase plays a very major role. Okay. Next one, the checkpoints of the cell. Already we have discussed. Let us see once again. So there are three major checkpoints in the cell cycle. One near the G1, a second at the G2 or M phase, nothing but the transition. Already we have discussed. Transitions are nothing but the checkpoints. And the third at the during metaphase, nothing but beginning of the metaphase. So positive regulator molecules allows the cell cycle to advance to the next stage. So if the checkpoint is been finding, yes, there is no mistake over here, no repair over here, no damage over here, it allows the chemicals and the substances to undergo the further actions until and unless the checkpoints are making confirmation, giving the confirmation towards the substances, so they will not undergo, they can't put their step forward into the next cell cycle. This is nothing but the checkpoints. So within this, in the G1 phase, cellular contents excluding the chromosomes are being duplicated with the help of a checkpoint, and each of 46 chromosomes are duplicated by the cell. Equal number of cells are being also maintained by the chemicals which are regulated by the checkpoints, and the cell double checks double checks, double time, twice or thrice they will be checking the duplicated chromosomes for error or making any needed repairs. After formation of duplicated chromosomes, are they responsible for forming a new cell again? Can they able to undergo the new mitotic cell division again or not? If they can't go, again they will take some time for repairing it, providing the energy for it and making the cell to undergo the mitotic or meiotic cell division. Okay. So, those are the roles played by the multiple checkpoints near the multiple stages. What are those? One near the G1, one more near the G2 or M, and third one during the metaphase, nothing but beginning of the metaphase. The checkpoints in the interphase will play a very major role in controlling the mitotic cell or meiosis cell division. Yes, and now let us see what is the purpose of the cell cycle. G1 checkpoint, the most important checkpoint, if cell goes through it, it will be a purpose of the cell cycle. The checkpoint near the G1, so if it is been done in a proper way, so then it is a purposeful cell cycle it seems. If it is not able to check, checking point is not checking it properly. So what is the use of the checkpoint near G1? There is no use. Okay. So for purpose of the cell cycle, the checkpoints must play a very major role. The most basic function of the cell cycle is to duplicate accurately the vast amount of DNA in the chromosomes. Okay, so the amount of DNA chromosomes, so the quantity will be maintained in a proper way, so that the number of the DNA, number of chromosomes also will be maintained in a exact accurate manner, and then the segregate of the copies precisely into two genetically identical daughter cells. The genetic material which is present in each and every chromosome, each and every cell must be maintained in a equal amount, not so high and not so less. Complete adequate amount of the chemicals or genetic information, genetic material must be present in each and every chromosome. That will be maintained by the checkpoints in the G1 phase, S phase, G2 phase and also the M phase. Okay. Next, see here the cell growth, the nucleus in the G1 phase, the cell grows within the cell body itself. Okay. So, and DNA checkpoint over here, after the DNA checkpoint only the DNA replication occurs, right. So, this is the process which occurs in the S phase, and again after DNA replication is been done, again there is a sequence after the replication of DNA, opening of DNA helical structure, single helical structure, again they will be binding. After binding, the sequence they will check. Okay, again there is a DNA damage checkpoint. Okay, so either the damage or the sequential order it might be different. So again they will check. After that the G2 phase occurs. Okay, so after the G2 phase again there is a checkpoint, DNA damage checkpoint first of all, and later DNA replication checkpoint. Okay, first of all it is functioning or not. After, if it is functioning, after replication, whether it did properly or not, replication is being done in a proper way or not is also being checked. And next one is the antiphase checkpoint. Okay, so what is antiphase checkpoint? Before the mitosis, the daughter cells have the capacity to divide mitotically, completely, interphase, prophase, metaphase, anaphase and telophase. So within these all phases, whether they could perform very well or not is being checked in the antiphase checkpoint. And next one near the M phase, spindle assembly checkpoint. Okay, so before going to the spindle fiber formation, so again in the synthesis phase we can interphase we can observe the checkpoint for the spindle assembly. Okay, so here we can observe the metaphase and the telophase, prophase, metaphase, anaphase and the telophase. So here we can observe the equatorial plate, and in the telophase we can observe the equal distribution. Okay, so now each and every after cytokinesis complete cells will be formed, two daughter cells will be formed by the single one. So here this daughter cell again undergoes the interphase, prophase, metaphase, anaphase and the telophase. And again the second divided daughter cell also undergoes the interphase, prophase, metaphase, anaphase and telophase cell divisions, and after that again they go for the continuous process. Okay, right? Thank you so much, dears. Hope you all enjoyed this video. We'll meet in the next session, right?